Thank you, Burkett. And thank you for hosting me here. I really enjoy coming here. This is actually the second time I'm in Munich. So, but I thought I'll just pinpoint where I'm from or where I'm located currently. So this is the campus of the University of Queensland. Just to give you a little bit of context where we are in the world. So this is on the east coast of Australia, and you probably know where Australia is in the world. So gives you an idea. We're just south of the Great Barrier Reef, which sits up here, and then some of you might have visited.
You have a unique perspective on Australia.
Sorry?
You show us Singapore. Everybody in the world knows Singapore, but nobody knows Australia.
No, not so sure about that. Okay. And then we'll go back to the presentation. Okay, so the topic of today is sequence motifs. Now, my understanding is that most of you have a computer science background. Is that right? Yeah. And I'd like to put this into a little bit of broader context and not just talk about biology. There is some interesting sort of method development issues involved in the three main tasks of sequence motifs, namely representing them so that we have an effective data structure, for matching them or finding them. So once we have a way of representing them, we want to quickly find occurrences of these things. And we'd like to discover them. So sometimes we have only partial information that will allow us to represent them. So we'd like to discover them from examples that might only give us an idea of what they look like. Does that make sense to you all, guys? I'd like to keep this interactive. If you think that I'm going too slowly, let me know. If you think I'm going too quickly, then the same. Okay? So you can imagine here we have a data set, and rather than think of this as biological data, you can think of these as a number of entries of emails, and you're tasked to flag occurrences of phrases that might indicate an imminent terrorist plot or something like that. And these are sort of emails and indices to the emails, and roughly give us an idea where these phrases might occur. But we don't know exactly what the phrases look like, although we might have an idea. They consist of several words, so it's a sequence of things that we're looking for. So as we scan those emails, we are going to make an assessment what strikes or comes out, or what might be overrepresented in this data. In actual fact, this is biological data, and most of the examples I'll talk about will be biological of nature. So I hope that you're okay with that, but I presume that you have some understanding of biological sequence data. And is it right that you've heard about sequence alignment and that stuff? Okay, good. So to come back to what this biological data is, I mean, it doesn't really make a lot of sense when you look at it, but you might guess that the first field that you see in this data file here has to do with chromosomes. And so CHR3 stands for chromosome 3, and then you have a big number and another big number, roughly in the same or very close to the first one, and then you have a couple of other numbers. We'll just bother with the first three there. So chromosome 3, and those two big numbers are actually two locations on chromosome 3. And chromosomes are big things, sequences of many, many positions. And between those two positions, there is something happening. This experimental technique that we've been using to generate this data file has said that here's something occurring. And that thing is occurring again on chromosome 2 between those two locations. And again, in a number of different, let's see, on a number of different chromosomes. And in fact, we're talking thousands or maybe even tens of thousands of locations here. Now, locations on a genome, and you should then think, well, that means that we're looking at DNA sequences, and we can quickly just use those locations to pull out the actual sequences that occur at those locations. We can look at them. And if you go back to the terrorist example I had earlier, where you're asked to scan emails that might be encrypted somehow, these are the codes. And in those codes, we'd like to find overrepresented or enriched phrases, if you like. If we think about the actual biological data here, we're talking about binding events. So on this DNA sequence, most probably a protein will actually bind. Although we don't know exactly where in that window where that protein binds. Typically, a protein would bind to a section which would be in the order of 8 to up to 20 nucleic acid or nucleotides long. So we're looking for a word, if you like, that would be in that order. Maybe it's over here. But it could be, in this sequence here, it could be over there. And in this one, at the end of it, we don't know. And it actually might not occur at all in that sequence. This is our DNA binding motif problem, and I'll come back to that and use it as an example. Primarily because it's an important problem, because we want to understand certain processes that are activated by these binding events. Partly because it's a simpler sequence to deal with. We're dealing with DNA sequences, and as you know, DNA sequences are sequences of only 4 letters as opposed to proteins, which would be 20. I'll be using or referring to examples in protein sequences as well. Does this make sense to everyone? And in fact, this sequence motif problem is relevant to quite a few different biological questions. And I'll be listing, or I have listed a few here, but there are many problems generally relating to data mining, so where we're trying to find information in large data sets. So some of the techniques that have worked for bioinformaticians to find the sequence motifs, like active sites in enzymes, like post-translational modification sites on these proteins that might be particular sequence sites that are being modified, perhaps phosphorylated, by enzymes. And often those enzymes will look up or be attracted to particular sequence motifs. Or it might be localization signals, and I believe, so Tanya, you would have talked about localization. Okay, so you would recognize this, and I have a few examples of that as well. Indeed, example 1, which will come up on the next slide, will talk about this. Or regulatory binding sites on DNA, which was the example that I started off with, and I'll come back to. So it's ubiquitous in biological sequence. We'd like to have methods that are effective to both represent, find them, and to discover them. Of course, our data sets will be invaluable to us, and some of those data sets will be very rigorous and rich in information. And we might have sort of detailed structural data that allows us to say exactly what kind of sequences that might make up these motifs, or it might just be partial. We have pointers that will give us ideas what the motifs look like. And sometimes we're simply just looking for words or sequences that are being statistically enriched. So it's a whole spectrum of different challenges that we're facing, and often we're somewhere in between. So we need methods that will help us with these problems. So protein localization is just one out of many examples where sequence motifs are key to understand how proteins translocate in a cell, and therefore help us to understand what they're doing, what partners they have, and so on, and what may go wrong. And I'll just pick a few examples here. This is a quite messy image, but it contains a lot of information and gives us a view of a range of different localization signals, including this one here. So we start off with a messenger RNA that is being translated into a protein sequence. It does so on ribosomes. Ribosomes, in this case here, when there is a so-called signal peptide at the N-terminus, so when this ribosome, which you see here, is starting to translate this amino acid sequence, the first bit of it, the N-terminus, if that contains a particular signal, it will associate with another protein or a complex called a signal recognition particle, which will help the ribosome to associate with a compartment in the cell called the endoplasmic reticulum. And the ribosome will actually continue to translate the amino acid sequence into that compartment. Of course, it's critical for that protein to have that particular signal, that signal peptide, for it to be translocated into the ER, the endoplasmic reticulum. So it's valuable for us to be able to identify what kind of sequences that would do just that. We can identify the subset of proteins that have that, and thus infer that these are proteins that will go into the ER and possibly continue along the so-called secretory pathway and be excreted or secreted from the cell. Now, so that's one example. Another one that I'll focus more on is so-called nuclear localization signals. So we're looking here at a different pathways. We have a protein being translated on free ribosomes, not associated with the ER, and they form this protein which folds up into a three-dimensional structure, and you know, of course, all about that. That three-dimensional structure, as it happens, might have a nuclear localization signal on its surface, which will allow it to interact with so-called importins. This is a general mechanism across many species. Most of the eukaryotic species would make use of the same kind of idea to have importins. So these are proteins that are going to associate with the protein and then carry it through the nuclear membrane into the nucleus, where it might perform a range of different functions. It might do things such as binding to DNA, which I referred to earlier, to make genes express. To understand that principle is thus important, and a starting point would be the actual nuclear localization signal and see what it looks like. This is just a long list of different localization signals, and I won't bore you with going through them in any detail. Instead, I'd just like to point out a few things about the mechanism behind nuclear import. So I mentioned that there are so-called importins, and one of them looks like this. So this is importin alpha. In fact, it has a partner called importin beta. And importin alpha plays the role of being an adapter protein, which means that it's actually responsible for interacting with the cargoes, those proteins that are going to be incorporated into the nucleus. If we look closer at the structure of this particular protein, we notice that there is a section of that structure that is highly conserved. And indeed, that is the patch of the important alpha that interacts with the carrier, or the cargo, I should say. And there are numerous of structures in PDB, the Protein Data Bank, which has this particular protein in complex with cargo proteins, or actually parts of cargo proteins. And they typically associate at that patch there. Now, those cargo proteins could, of course, be a range of different proteins. And as I mentioned earlier, they might have a function in binding to DNA. They might carry out completely different functions as well. So these are proteins that are widely different. So if we wanted to sort of understand what makes them being imported into the nucleus, we can't rely on something like sequence alignment, because they will only have a little bit of them that are similar to other proteins that are also imported. And that little bit is what we see interact with this patch here. So most of the proteins will interact primarily with this section here. So if you look closer at this schematic view of the protein, or the important alpha, you can see a number of alpha helices lying there. And they form this kind of pocket that allow it to sort of wrap around a region of the cargo protein. And most of them would associate primarily with that section there. And if we take those proteins and we identify what kind of amino acids that are placed there, they are typically basic residues. So things such as lysine and arginines, but not always. There are also some cargo proteins that not only associate with that section, but they also associate with this site over here, which is referred to as the minor site. And there we have another section of basic residues. So this is referred to as the monopartite nuclear localization signal, and this is the bipartite, and you can probably guess why. Okay. Of course, we haven't really said how we sort of came up with those sequence signals, and I'll come back to that. Here's coming back to that DNA binding problem we talked about earlier. So you guys know about gene expression? Yeah, so genes being expressed in a cell, and that is often caused by other events, regulatory events. And some of them would be modulated by so-called transcription factors. This is a class of proteins that all bind to DNA either directly or indirectly. And I think, okay, so they would typically bind in this region here, which is just upstream of a gene on the genome. So here we have the gene. This thing here, the RNA polymerase, is going to transcribe this gene sequence, and it will result in an RNA molecule. But binding events just upstream of that, potentially located at other sites that are sort of proximal in space, not necessarily in sequence. So this thing over here, this binding event, might be way upstream or possibly way downstream of the gene. And the file that I showed you initially was a file with these binding sites for one particular transcription factor. So there is a limited number of transcription factors. There are probably in the order of thousands in the human genome. And they seem to have different roles to play. Some of them work together, some of them work individually. But they will all have sort of this kind of influence on the expression of genes. In some cases, they will repress the expression of genes, so that means that the output here will be lower. Some of them will excite expression or increase the rate of expression. So it's a logical circuit, really, that we're dealing with here. So to understand what makes them bind to DNA is going to help us to unravel that sort of regulatory logical circuit. Okay, and here's an image of, this case, the transcription factor binding to DNA. So if you want to understand the binding site, it means that we have to look at what nucleotides that we have here on the DNA. Okay, and these transcription factors will have preferences. They like to bind to certain DNA sequences. Some of them will have very specific requirements or preferences. Some of them will be more general. Okay, so when we construct these motifs, it could be done by hand. So we might have very detailed, rich information. And I'll exemplify this in just a minute. It could be done from sort of consensus, where we have information, perhaps in the form of a multiple alignment of many of these, say, binding sites or localization signals or whatever you have. Or it could be done through automated discovery. The use of statistics here is key. Or machine learning methods. And statistics will help us to understand what is overrepresented or underrepresented, for that matter. And machine learning techniques will help us to deal with data where we have labels assigned to our data points. Okay, so here's an example where we have more detailed information. So here's another transcription factor. Indeed, one, we have a couple of so-called zinc finger domains that are being associated with DNA, and they like to bind to particular DNA sequences. So we might, when we look at that DNA sequence there, find this word. And that would be detailed information. We need many of these structures in order to generalize that, because if you only look for that particular sequence, we might in fact not understand that it doesn't really care about what letter that we see there, or perhaps this one here can be changed to a T without any concern to the transcription factor. So we need many more examples to understand the degenerativity, the degenerate nature of these binding motifs. So, and here's another example where we think in terms of finding a consensus. Here we use an experimental technique called SELEX, which is going to allow us to look for what does DNA sequences, what DNA sequences are being preferred by a nominated transcription factor. So in this case, GATA3 is the transcription factor, and SELEX will generate lots of random peptides, or I should say random nucleotide sequences, and then identify a subset of them which bind to this transcription factor. So here's a bunch of them from one experiment, and our task then would be to identify what's common amongst all of these, because they all bind to that transcription factor. Okay? But clearly they're not identical, so there must be more to it. So you should probably be thinking now, well, let's use sequence alignment. And indeed, we can, because these sequences are relatively short. So if we perform sequence alignment, not allowing any gaps to occur, we're seeing this. And if we look closely at the section of this alignment that is sort of conserved, or which is shared amongst the different sequences here, we do indeed find a common pattern here. So we see GATAA in quite a few of these sequences. So we can use sequence alignment to do this. So what do we do next? We're going to identify some kind of descriptor here, or a representation of that motif, so that we can then scan DNA sequences to identify potential binding sites. So there are a number of things on this slide here, and I'll take you through them. So the first thing I'd like to sort of point out here, that we can start with the counts. So if we look at the different nucleic acids that are found in that alignment, say in position 10. So if we look back here, so position 10 would be somewhere along there, okay? If we count them, we'll end up with these counts here. In fact, I'm glossing over one little detail here. That is that we have gaps in this alignment. What do we do with them? The simple thing would be to just ignore those sequences. But what would happen to say, if we look at the first column here, we would just have an A there and a C there. Okay, so there are only two possible symbols according to that alignment that would be allowed if we were to create a representation from this. That should sort of ring alarm bells to you, because that means that we're going to require A or a C in that particular position. So if we were using such counts, then we'd be in trouble. Of course, we can limit our alignment to just the section which is highly conserved and just do the counting there. Yep?
Some question. Why don't you put a number at the end of the sequence which tells you for how many places is it, should we align it left or right?
Yes.
Like minus or plus?
Yes, so you could add information like that, and in fact, let me just go through the next step here. So what we're doing currently, or what I've done here, is to choose to actually look at the gap and say, well, the gap could mean anything. It's a wild card, basically. And so we would end up with a count here for the first column, which would say 15 and 15 for A and C, and then 14 for G and T. So basically we see a small preference for A and C, but we're rather uncertain as to what should go in that particular column. The alternative would be just to count A1s and C1s and then 0 for those, but that would mean that we would accept no sort of matches with anything that has a G or a T there. Of course, we could weigh that column differently from the others where we have more data. Now, and we will come back to this particular issue. The other matrix that we see here is one which contains frequencies, and those frequencies are based on the counts that I just mentioned here. So we can determine the preferences for different letters here in terms of frequencies. So for position 10 or column 10, we would prefer to see an A, but we're not that certain. It's easy to calculate this, and we could use this as a scoring matrix if we like. Now the third thing that I'd like to mention here is this illustration here, which you saw an example of earlier when it came to the nuclear localization signals. Here we're dealing with just DNA sequence, so we have 4 different symbols. And you can probably see that this is a relatively meaningful visualization of what is contained in that alignment here. Have you already learned how to make these sequence logos that I'm showing here? Do you know how to do it? No? Okay, so I should tell you, because these are really popular in the bioinformatics literature, and if you're reading a bioinformatics paper, it's quite probable that you'll see some of these things, and biologists love them too, I should say, because they give you that sense of what the sequence pattern looks like without the numbers, if you like. But of course it's based on numbers, and it's a quite clever way of visualizing sequences. So, of course, what you see on the x-axis here is the position in the alignment or the matrix. And then on the y-axis here, you see something that is expressed in terms of bits. Now, you're a computer scientist, so you would know everything about entropy, right? Or information entropy? Yeah, so this will be easy to you. First, frequencies are easy to determine. We just take the number of a particular letter that we see. So this is an index that I use for the particular identity of the letter, and then there is an index for the position in the alignment. And then we're going to divide that with the total number of sequences. So we end up with a matrix like this, or like the one that we had on the previous page. And this is the sequence that I'll be using in the next example here. It has 20 sequences, so yes, if you count the number of A's that we see in the first column here, so 1, 2, 3, 4, 5, you end up with 0.25. Okay, so how do we come up with this thing that illustrates the sequence that we see in that alignment, or in that set of sequences? Okay, it's going to be a couple of things involved here. We first look at the height of every position in the sequence. So you can see that these are definitely higher than these, and what that means is really that this is more certain information. We're pretty confident that we're dealing with a particular symbol here. In this case here, there is an assortment of them. We don't quite know. And that's based on the Shannon entropy, that height of the letters, total height. And in fact, it's this. Okay, so the information in a particular column here is this value. Now, log 2 of the size of the alphabet. So if we're dealing with DNA sequences, it's 4. So log 2 of 4, you know that? You can do that in your head, right? 2? Yeah. In fact, that's why we see the columns with the greatest certainty reaching all the way up to 2 there. Yeah.
So I have a question. When you count the frequency of residues, do you count the gaps? I mean, if I have 10 positions and the A occurs in the bonds, so what is the frequency of this residue?
So that's optional whether you want to sort of include the gaps. In this, I think that, well, in this case here, we weren't dealing with gaps at all. I'm not sure whether there is a default way of doing this. So this is using a tool called the web logo, which you can find if you go to that URL, whether that deals with the gaps in a particular way, but it would be optional. So if we sort of decompose this expression here, so just bear with me for a minute. So we have that maximum value of 2 here, and then we're going to deduct the entropy, the Shannon entropy for that particular position. Okay, so that's how we calculate the entropy, which is just this standard formula here. So when we're dealing with I mean, since you know how to calculate entropy, I won't dwell on this. Okay, so if we have a big sort of entropy, that means that we're going to deduct everything from that value 2 here. So we'll end up with 0. So when basically the letters are random, like it was for the first position. So if we go back to this, you'll see that we had 5 A's, and then there were 5 T's, and then wasn't quite random with the C's and G's, but it's very uncertain what we see in that particular column. It actually reduces, let's say it's 8. I just want to verify that we're talking about I've calculated this for the right alignment, yes. So it'll actually be so small, or the Shannon entropy will be so big that we can't see the letters here. Okay. Okay, so we know now how to calculate the total height of each position, and then we're just going to take the frequency of a specific letter and multiply that with our total height there. So that means that in this case here, the second column here, G was the most frequent nucleic acid, so therefore it has also the greatest proportional height. Does this make sense to you? Yeah. So coming back to the gaps here, I mean the upshot here would be that if we have all gaps in a column, we would be completely uncertain. Okay, so we should be looking at something like this, and we would get that result whatever way we dealt with this, because we would either count everything would be would be a uniform probability. To come back to what WebLogo does, I'm not entirely sure, but I hope you get the gist of the idea.
Well, why don't we just treat them as additional letter in the alphabet? Like instead of 4, it's
well, we don't want to. I mean, the gap itself is the absence of information. It doesn't actually give us any information. So I think it would be dangerous if we were to construct scoring models that would use the gap as something to look for. It's better to sort of be open-minded about what might be found there. Have you talked about hidden Markov models at all in the class?
I mentioned them, yes.
Okay. So the types of methods that we're dealing with here, we'll look at the columns, and then we'll look at the columns independently. So we would do counts for each of the positions here, and we would count only the letters that occur in there. Now, when it comes to gaps, there are some benefits of looking at the context of the symbol, and also more generally. And I might make some comments about that a little bit later in the material, in the value of looking at the windows of these letters. And those windows will allow us to treat gaps differently. Hidden Markov models would be a direct way of allowing us to work with sequences that have different lengths. Okay, so I may come back to that a little bit later. Okay, so this is just to help me remember that I'm going to talk about proteins now again. So now we're dealing with 20 symbols instead of 4. The idea of sequence logos can be applied to 20 symbols, no problem. So when it comes to these nuclear localization signals, what we can do is to dig up information about the proteins that are known to be translocated or located in the nucleus. So there are experimental techniques that will allow us to assess where a protein is. Although they're not perfect, and sometimes a protein might go to one location, and in another condition it might go to another. And sometimes they're going to have dual locations, so we see some proteins going there and some proteins going to a different place. But anyway, in some cases we would have reasonably good information of where they're located, and if they're in the nucleus, we would expect them to have a means to get there. And some of those proteins, at least, would have a so-called nuclear localization signal. So if we were to put together a data set to construct a motif for nuclear localization, we'd be doing database searches where people have identified where that, well, first of all, the compartment, but also where that sequence signal is. So if you have an idea what kind of sequences that work, you might identify the site in your favorite protein, and you could mutate perhaps that site so that you can assess whether that was functional, whether that was responsible for that protein to enter into the nucleus or not. So that's why people are able to do this with some certainty. So in this case here, it's a nuclear localization signal that is found in position 23 of that protein up to position 28, and I've marked that in red here. Okay, so we could in fact construct an alignment now directly from that kind of information. So we basically pile up all the sequences that have that specific annotation. So it could look something like this. I should say for accuracy that this is a different type of data set, one that was generated experimentally, not unlike the one that I mentioned earlier for DNA binding sequences, where we have random peptides which were associated, according to the experiment, with importin alpha. So we put them sort of on top of one another, possibly fixed it up to make sure that everything aligned neatly, and then we can construct scoring matrices, just like we did previously. So we'll just look at this as an example where we're dealing with amino acid sequences instead of DNA sequences. So we have our counts. I've just included some of the amino acids here because 20 would just be too many. And you can see that the counts for lysine and arginine are much bigger than for the other amino acids. Now, this column here, can you guess what that means? So 0.06, 0.06, 0.09, 0.09, 0.06, and they align with the amino acid that you see there. Can you guess what that might be? If I tell you that this is the frequency at which that amino acid occur in some really big data set. So in 6% of all positions in this big protein set, we see a lysine. And we see a serine in 9% of those positions. So this is a background. And that background will help us to assess the surprise that we see a particular symbol in a position. Okay? This will indeed help us to come up with a better score. So have you talked about substitution matrices? So you know about the log odds ratio and all that. So
some of the people in the room do, some don't.
Okay.
That was last semester.
Okay. I have a formula later, so I'll just flag the background here as being a valuable piece of information for us. And this would go for if you're interested in text mining, for instance, and you would be looking in many documents, and you would see, and you look for words or phrases composed of words. You might have so-called stop words, or words that you don't want to look at. You don't want to include them in your assessment because they're just so common. Mm-hmm. This is the same idea that you don't want to sort of be too biased in your searching and your discovery by something that is occurring just so often that it would occur just by chance. Okay.
Using, for example, a hidden Markov model, would these be the initial probabilities?
Could be, yeah, could be. Okay, so I'm going to take that idea further, but I'd like to, while I have this slide on the screen, say something about an alternative way of representing motifs that people have been using quite a bit. I'm not sure whether you can see it very well, but in this sort of lower end here of the diagram, we see two example regular expressions. And as computer scientists, you would be well aware what a regular expression is. So you would construct these discrete representation of what is a match to our motif or our sort of alignment of interest. So that can of course be made very general. So we would include all of the letters that occur in a particular column. So in this case here, let's say it's 1, 2, 3, 4, 5, 6 positions. So it's actually over this whole alignment here. And it says that we should allow R and G to occur in the first column. Now that will not match all of the sequences here. So we might want to include more letters here, but that would also mean that when we use this as a search pattern, it will have much more or many more matches, and some of which will be just not right. So we want to balance the specificity, that is that we only want the sequences that are real to be matched to our pattern, and the sensitivities, we want to include as many of the real matches as possible. Okay, so we come up with a description like this. This is a shorter version which only looks at the positions that are highly conserved. But these are just example regular expressions that we can use for searching. But they would be discrete, so either you match or you don't. Whereas the probabilistic representation that we'll be using here, on basis of the counts, will be able to score sites differently. So there would be a spectrum of scores that we can achieve. And some of the sequences will have a greater score just because they're more alike the patterns that we use to construct the matrix. In the end, once we've included these background frequencies, we get a scoring matrix like this, and I'll show you how to come up with this shortly. And here's the sequence logo. Okay, so when we want to use this for scoring or for scanning sequences, we need to be more careful than what we've been in the past here. So plain frequencies will fail sometimes to capture the element of surprise. Alignments are not always obvious or even accessible. We need a stronger theoretical framework to talk about scoring, and then later discovery. So that's what I'll talk about now. You need to stop me when we're running out of time here. So do you usually take a break at some point?
No, yes, just before the end of it.
And
you have about 20 minutes to go or something.
Yeah, okay. All right. So I'll talk about probabilistic models here, and this is essentially a system that simulates the object or process under consideration. Sounds very vague. It has parameters which we will tune. We will change them to infer or assign different probabilities to possible outcomes. So here's an example. We have a process like a roll of a die. Okay, and we have a number of different outcomes, and we have parameters, and these would indicate the probabilities of these 6 outcomes, either directly or indirectly, depending on what the model looks like. In the simplest case, we might have a probability associated with each of those outcomes, but there might be some more complicated scenarios that we'd like to consider. Probably not for a die, but anyways. Of course, when we're dealing with probabilities, we have a number of constraints to fulfill, that they need to add up to 1, for instance, and they're not going to be negative ever. We have inference. We can implement it in a variety of different ways, and the outcome would be something like this. So we would assign a probability to the outcome 1, which would be 1/6. And there would be a number of different rules of probability, and I'll just look at some of them here because we will benefit from them when we talk about motif discovery, and I think I will be needing some time on Thursday if I'm going to get through the discovery part. So bear with me. Joint probability, this is the probability of x and y. Okay, it looks like this. You would know this, right? Conditional probability looks like this. So we have the probability of x given y. When we're talking about joint probabilities, okay, so joint probabilities of x and y specifies the probability of each complete assignment of values for both x and y. So, and we can have this for many variables. Sure. So if I write here, it'll end up on the video as well. Yeah.
Is it? It always works good enough. When I write on the whiteboard, I've never looked at the videos.
I don't really exactly remember, but
I think you can see our heads too.
Okay. Well, anyway, I'll just explain it in words then. So if we have...
I do it for your information. I do it all the time.
Yeah, okay.
And there's an advantage for the ones who are sitting in the room. So.
Okay. Okay. Okay, but I probably have to use a pen that works. Okay, so we have two variables, x and y, and let's pretend that these are nucleic acids, so they can take a value. Or maybe even just true or false. Okay, so the variables, these random variables, x and y, they can either be true or false. So our joint probability can be drawn as a table in the case of discrete random variables like this. And we would assign a probability to each atomic event, that is every possible assignment of the variables in the joint probability. So it might be something like this. Sorry. Okay. If I include a third variable here, so I have a z here too, well, this table will just be twice as long. And our probabilities here on the right-hand side would still add up to 1, so we have to distribute it even further. Okay. We can go the other way, and we can do so-called marginalization. We can sort of go from one joint probability, which consists of 3 variables, and to the case of 2 variables, just by summing out. So if I wanted to go from xy to just x, I just have 2 possible values, and I would take these 2 values, 0.4 against the sum of those 2. So 0.4, 0.6, okay? Because x is true there, x is false there. That's essentially what we're doing here, okay? And then we have the conditional probability, which is probably the more exciting of them. But I should also note that the joint probability contains all the information that we need about these variables. We can pull out any information from there. And this is actually one example of how we can do that. We can represent it as a conditional probability. So we can calculate it by taking the joint probability and dividing it by the marginal probability of one or more of the variables. So we can write it like that. And of course, so that this is the same expression as we saw on the previous slide. And we can just rearrange the expressions here and get this, this formula, which expresses the joint probability in terms of the conditional probabilities. And this is often referred to as the product rule. And of course, since this thing is reflexive, so we can turn those 2 things around, whereas in that case we can't. We can derive the so-called Bayes rule, and you would have seen this, right? It's not the first time you see Bayes rule. Where we can turn, or we can, if we have information about one conditional probability, we can in fact determine the the other conditional probability with those two variables swapped around. Once we have better information about the prior probabilities of those two variables there. This is advantageous to us. We'll come back to that. So first we'll talk about sequence models. So now we're going to look at not Boolean events like this. We're going to extend it to sequence data, or where we see a number of positions. We see symbols that follow one another. So biological sequences, DNA, RNA, or proteins, doesn't matter, or if you prefer text, natural language, English, or German, can be thought of as outcomes of random processes. You have a monkey typing on a typewriter and producing the works of Shakespeare, okay? With a very small probability, but it can happen. It makes sense to model sequences using generative probabilistic models. So we think about our model as being able to generate these texts or these sequences. So basically, we are going to design that monkey that types the works of Shakespeare here. And of course, if we can do that, so we make sure that the monkey types the works of Shakespeare every time, that would be a model of Shakespeare, or the works of Shakespeare. You can think of a probabilistic sequence model as a machine that randomly generates sequences. Okay. So now we're getting into the problem of discovery here. So we're going to find a model that will represent something that would have generated sequences in the first place. Does that make sense? So if you think about these nuclear localization signals, I mean, at some point those sequences would have originated. How did they? What sort of enforces those sequence signals to be functional? And we'd like to find a model for that. So parameters can be estimated from sets of trusted examples, and we're going to refer to that as a training set, or D. And you've been looking at machine learning, right? So you would be familiar with this way of thinking about finding models. And with large sets of fully observable examples, we expect measured frequencies to be reasonable estimates of underlying probability. So if we have big enough data, we think that is going to accurately represent the real world. So, I mean, our data will never be complete, probably. But having a big enough data set, it's going to be a good enough estimate for us. So a model with parameters theta and a set of data, that's what we're going to deal with here. So theta is the symbol I use for the parameters. The maximum likelihood estimate for theta is the value that maximizes this expression here. So the probability of D, seeing D, given theta. So this is our model. It's going to generate some data for us. So this is our monkey typing on the typewriter, and this is the text that it's producing. And we'd like to have a monkey that will generate exactly the kind of text that we provided to it. If it's doing that well, surely it must have been, it encapsulates everything there is about that process that generated it in the first place. So here's the simplest possible example. So we're dealing with, again, a die, in this case a tetrahedral, a four-sided die. So it would look something like this in the two-dimensional render. So on each of the sides, you'd find one of the four letters, A, C, G, or T. Now, if you roll this die 100 times and you write down the letters that come up, or down, because obviously this tetrahedral die will sort of not show a letter on the top, but at the bottom. And you can find the frequencies of the outcomes, and these would be our maximum likelihood estimates. And in fact, so our die here is the real process that we're trying to model. So if you just throw that or roll that die sufficient number of times, we'll get accurate estimates of the model parameters. And that will give us the greatest possible chance of taking those, or to implement this as a computer program, to generate the same sequences that we would get if we were to roll the die. You agree? Okay. So if you think about this in terms of the biological sequences again, so here's a sequence that I've just typed in, but it could be biologically meaningful. It's probably, could it be 100 positions long or so? And I'm using some Python code here. I will give you a URL at the end where you can download this Python code if you wanted to play with these examples. But I just wanted to illustrate what the basic steps here involved. So we have the sequence, and I'm going to create a model which is just going to be this simple distribution over the possible letters that we have in our alphabet, DNA alphabet. And I'm going to look at each possible symbol here, and I'm going to observe it. And then at the end, I'm going to print out what the model looks like. So this will give us the parameter values. So you can see here, A is 0.21, C is 0.27. You can probably guess that these are the probabilities. So what do you think happens in here? Is it doing anything complicated?
Just probably observes changes on the image.
It's just counting your symbols. So for everyone here, we'll just say, okay, I'll have a C, and then a T. It will just keep track of the counts here. And in the end, it will say, okay, I've seen 100 symbols or whatever, and it will figure out the parameters from there. I guess that if we stopped earlier, maybe if we stopped there, the probability of seeing a C would be 50%. Okay? So this is the result of just looking at those symbols and counting them. That's the maximum likelihood estimate of the parameters. So that means that we can use that model to generate our data, or to generate new data, which should look like the data that we provided as training, right? So here we have sequence 2, which is going to be an empty string, but I'm going to populate it. I'm just going to concatenate the guesses that my model is making. So I'm going to generate a symbol here. I'm going to sample basically that probability distribution. So I'm producing this sequence here. And for your convenience, this is the sequence that we produced as training, or this is the training data, and it looks like nothing like this sequence here. Maybe there are some similarities. Indeed, we can assess some similarity just by looking at or using this as training data and see what the distribution looks like. And indeed, the frequency or the probability of A is the same as it was in the training data. And C is not too far off either. G, similarly, close, and T.
The problem is that the conditional probabilities are not the same. So if you have AA, I mean for the single nucleotide it is, but for example C and T appears much more frequently than, and it's not calculated in models.
Yes. So what should we do?
So also count the conditional probabilities and let's say stop at n words, like I don't know how far you go. Stop it and recalculate the model.
It's a good idea, right? So you would look at windows of these things here. So you would capture some of the dependencies between the different positions, right? And ideally, what we'd like to do is to count, or to look at, to go back to the original sequence here, to look at this whole thing and memorize that and say, this is what the sequence should look like, right? That means that we can replicate it easily, but there would just be no ability of such a model to generalize. So the more power you give to the model in terms of number of parameters and the types of relationships it can model, it can benefit in terms of the ability to reproduce the actual training data, but it will lack in ability to generalize, to go beyond. Perhaps there is a section in this sequence that is completely irrelevant. So there is that balance that we need to consider. So too powerful model is not necessarily a good thing. And of course, if we look at windows here, we're going to deal with a much larger space of parameters. And we might require more training data in order to find what would be the most appropriate parameters. So the number of things that should be sort of considered here if you think about this as a data mining problem or a machine learning problem or a statistical problem. For now, we'll actually be happy with this very simplistic model. And we're going to think of each column in our motif as one of these sequences. And of course, we don't care in which order the sequences in our alignment occur. So it would be irrelevant to sort of capture that kind of thing. So, position-specific probability matrix, and we're dealing with frequencies here again. Sorry to change the notation. So this is the probability of a residue A at position U. So previously we called this F, I think. Now it's Q. This is a particular binding site that I've taken from a database, but it serves to illustrate a couple of points in how we should be constructing a scoring system. Okay, so we have observed binding sites, and in fact they come mostly from so-called SELEX data. So it's that sort of chunk of relatively random nucleotide sequences that we align, and then we form this kind of count matrix. So that's where we start. And I've put it up here on top again. So in the first column we see lots of A's and a few examples of the other three nucleotides. The first thing I'll do is to do something called add pseudo counts. Guess why? So what does that mean, first of all? So I'm in this column here, column 2, I have 59 A's and none of the other letters there. I'm going to add 1 to each of those letters.
I think the problem here is when we consider the 0 in a probability calculation, it will make the whole probability 0, and
yes,
using the 1
yes,
can protect from
yeah, you're right. So we're going to smooth the model quite arbitrarily here to just say, well, in the absence, I mean, the extreme example would be that we have no data. In this case, we have reasonably big data. So we have 59 counts, so 59 example sequences, but let's pretend that we only had 5. In which case, it would be the effect would be quite dramatic if we only included A there, so let's say 5 counts and nothing there, because we have such a small sample to make any inferences of parameters from. So it's a good idea to introduce this background frequency. It's like a prior, if you like. So we do that. That's it's a general idea that people use when they come up with these scoring methods. And from there, we move to a frequency matrix. We know how to calculate that. And then we come to these background frequencies that I talked about earlier. So in the case of nucleotides, they're relatively evenly distributed, but not quite. And this distribution will actually differ from organism to organism. So it could be critical to actually model this background, because we will then be able to look at the frequencies in the context of these. So we are less surprised to see a G just because it's occurring by random. So when we see it in an actual hit or match, then we will not be as surprised as we would if we see, say, a T there. Okay. So we're going to take the foreground probability divided by the background probability. So if you think, well, this is a match, this is a background with even, so sort of think that it might be either way, we'll end up with a ratio of 1. So let's see if we can find one here. They're all sort of pretty far from 1. But that would be where we have no idea really. It could be an actual match, or it could be just a chance match. But some of them will have a bigger value than 1, and some of them will be having a value which is close to 0. So what we typically do is just do the log of that, because our ratio will be, if you think in terms of how values will be distributed, it will never, of course, be exactly 0 or below 0. And then you will have values at 1, and then you can have infinitely big values. So it's a very odd-shaped distribution or density. So a better choice is to do the log transform of that, and you make sure that this thing is centered on 0. And then you can have negative values, which would indicate that your ratio is closer to 0. And the positive values indicating that you have something that is above 1. So the final thing where I finish before, well, I'll talk about motif discovery, if okay, on Thursday, because I think you have another lecture then, right? So scoring is based on that kind of matrix. So the final thing that I ended up with here, that is going to be our scoring matrix. So just if you've seen substitution matrices, they will look very similar to this thing here, and they're going to be used in a similar way. So when we look at sequences, or in a particular position of a sequence, in this case here, I'll take this scoring matrix, and I'm going to take the values, which is these log transform values that we see in the matrix. I'm going to identify the position in that matrix which aligns with, or that is appropriate for the symbol. So C in this case would be the second element in the first column. This G here, since it's the second symbol in the sequence, will be the second column in this scoring matrix, and G would be the appropriate row there. So I've indicated what we're dealing with here. So when we look at 5 residues, or sorry, nucleotides in the sequence that we're searching through, this is the window that we're referring to, with highlighted here. And we're just going to sum these, and the reason, well, we sum them because we have log transformed our odds ratios, so that would be the appropriate way of combining their scores. If we hadn't done that log transform, we would have to take products, but now we're summing just as we would with a substitution matrix, and we would have a score minus 12 for that particular position, or for this window of the sequence. And we continue, look at the next position. So same matrix, it's just that I've highlighted different elements of that matrix. our score is -11. And here's one which is positive. So we have, again, same matrix, different highlighted elements. We sum them up and we get a positive score here. So that's probably a good match. And if we look at this, so we have TA, double TA, it's probably not too dissimilar from this thing here. So indeed, the first one is not right, but the other 4 would be. So that's how we get our sort of scores for each of the positions there, and we would slide across the sequence to determine where we have something that looks reasonably high for us to make a call. Of course, we make decisions in regards to what would be the appropriate threshold. Yes?
Could you go back a couple of slides, please? Could you explain again how you are highlighting this?
Okay, so we're now looking at A as the first symbol, and T as the second, T as the third, A and C as the fourth and fifth respectively. And they would correspond to the highlighted elements here. So you'd have A, T, T, A, C. Okay?
5 is a random number, right? So you just chose 5.
So the reason why I picked 5 as the window size here is because those were that was the width of the matrix for which we had data to construct our scoring matrix. Okay. Next up, I'll talk about motif discovery, but I'll do that on Thursday if you're okay. That's the most interesting bit. I've been going through quite a lot of boring stuff, but motif discovery involves some algorithms. We'll be talking about so-called Gibbs sampling, which is a very general method that you can use to find probabilistic models for many types of data sets. So I hope you can take something general out of that. So I hope to see you on Thursday.